Everyone. So we are going to continue our discussion on carbohydrates. So in the previous class we have discussed about oligosaccharides, and today we'll continue with polysaccharides. I think I have stopped somewhere here. Polysaccharides are types of carbohydrates which are formed of many monosaccharide units, and the number of saccharide units or sugar units might vary from anywhere from 10 to thousands. Okay, and these polysaccharides are also called as glycans, and there are actually two types of polysaccharides, and they are homopolysaccharide and heteropolysaccharide. Now first let's see what are these homopolysaccharide. Homopolysaccharide, as the name itself says, homo means similar, saccharide means sugars, polysaccharide many sugar units. So homopolysaccharide means carbohydrates that are formed of similar monosaccharides. Example: starch, glycogen, cellulose and chitin. So all these are homopolysaccharides because basically they are formed of similar repetitive monosaccharide unit. That means when you hydrolyze them, you are going to get only one type of monosaccharide unit. Okay, so homopolysaccharides are those type of polysaccharides which are formed of only one type of monosaccharide units, and on hydrolysis they give only one type of monosaccharide. Okay, first let's see starch. This is an example for homopolysaccharide because it is formed of one type of monosaccharide. Now what is the type of monosaccharide that forms starch? It is glucose. So we can take example of starch, glycogen or cellulose. All three are formed of a similar type of monosaccharide unit, that is these three are polymers of glucose. Okay, and here this starch, it is a long chain of glucose, not just only single chain, it is formed of multiple chains. Okay, each starch molecule is formed of two types of polymers. One is called amylose, the other one is amylopectin. Okay, and here these two types of polymers are slightly different from each other because this amylose, okay, this amylose, it is formed of unbranched chain of D-glucose connected by 1,4 linkages. Whereas, see here you can see, okay, you are going to see amylose here, okay. See, this is a long chain of glucose molecule formed of glucose. See, this is one glucose, this is one glucose, second, third, like this on both the sides it continues. And remember, we have discussed that two glucose units are connected by a glycosidic linkage. And here observe carefully, see the linkage between these two, okay, this and this is called alpha 1,4. The reason for this is here, see, this is the first carbon atom and this is the fourth carbon atom of this glucose. Now what happens when the glycosidic linkage is formed between the first and the fourth carbon atom of two different molecules? The linkage is called alpha 1,4 linkage. Okay, so in amylose the type of linkage you find is alpha 1,4 linkage. If and now why it is alpha? Because both of these are alpha glucoses. Okay, because of that this linkage is called as alpha 1,4 linkage. So in amylose you find amylose is a unbranched structure. Okay, it is a unbranched polymer of glucose units. So here you see that glucose units are connected by alpha 1,4 linkages. It is a linear chain. No branching is seen in case of amylose. Whereas in case amylopectin, okay, amylopectin is something that is similar to amylose because it is also formed of these branches, these linear glucose molecules. In addition to that, it also has these branching. Okay, so you will find, in addition to the linear chain, that is seen, so like this, in addition to the linear chain, you will also find that somehow they are connected by branches. And what kind of branching is seen? It is alpha 1,6 branching. Now what is this 1,6? 1 is the first carbon atom of this particular glucose molecule, and OH from 6th carbon atom of the branched type. Okay, so let us say that this is a linear chain, on both the sides it runs, and let me just take another color here. Okay, so this is the linear chain on both the sides running. Okay, and here is our glucose molecule, and this is the 6th carbon atom. Okay, this is the 6th carbon atom. Now what happens here? This 6th one is CH2OH. Okay, it is CH2OH molecule, and here and in this, the first carbon atom of this particular glucose molecule has OH here. Now what happens? A molecule of water is eliminated, so that now this O will form a bond here. Okay, it will form a bond here in such a way that first carbon atom and the 6th carbon atom are involved in forming a glycosidic linkage, and that is why this type of linkage is called as alpha 1,6 linkage. Okay, so remember, in case of amylose, it is the linear chain of glucose units. Amylopectin, the second polymer of the starch, it has a linear chain, plus it also shows branching, and the branching is between first and the 6th carbon atoms of the two glucose units. And again, once, see, this branching does not mean that it will stop here. Next, this continues here on this side. Okay, so it continues on this side. You can see the chain branches are shown here. So amylose and amylopectin together, they form the starch molecule. But on hydrolysis, they are going to give you glucose molecules only. Okay, so now usually we have certain points here to remember. So it is its molecular weight is higher compared to amylose. Okay, and it is highly branched. Remember, amylose is unbranched, amylopectin is highly branched. In amylose, you find 1,4 linkages. In case of amylopectin, in addition to 1,4, there is also 1,6 linkage. And so how frequently the branches occur? At every 24 to 30 residues, okay, branching occurs. So like this, if the glucose molecules are present, 24, anywhere between 24th and the 26th, another branching takes place like this. Okay, so that's about amylose and amylopectin, or in other words, structure of starch. So starch is a storage polymer of plants. Okay, and what is its use? Starch is used to get the food. So like you eat rice, okay, or you eat anything from the plant source, potato for example, sweet potato, carrot, or whatever it is, the cells will contain the starch granules in them, and when you consume that, your body is going to get the starch. And if you recall, in third semester you have studied the digestion of carbohydrates, where you have come across digestion of starch in mouth as well as in the intestine. And if you recall, alpha amylases will break the starch. Okay, amylose and amylopectin both will be broken down by alpha amylases, and again recall, they can break alpha linkages, alpha 1,4 linkages. So whenever there is a alpha 1,6 linkage, that will not be broken by the salivary amylase. So the significance of starch is that it acts like a good source of energy, because it is going to provide a large number of glucose molecules to you, okay, so that you can get, your body can generate energy. Next, glycogen, next type of carbohydrate which we will be, the polysaccharide we will be discussing is glycogen. So this glycogen is the similar, it is similar to starch, but it is a polymer of animals. So that means animals store food in the form of glycogen. Plants store carbohydrate in the form of starch granules, and animals store the glucose or carbohydrates in the form of glycogen. Okay, so here glycogen, it is a storage polysaccharide of animal cells. It is also formed of glucose subunits. Okay, now the linkage is again similar to amylose. Here also alpha 1,4 linkages are present between glucose units and 1/6 linked branches are found or also found, but branching is more profuse than in case of starch. So remember, there for every 24 to 30 residues you find branching, but here for every 8 to 12 residues you are going to see the branching. Okay, so here it is more like amylopectin, linear chain of glucose, plus there will be branching too, and the branching is between with the range of 8 to 12 residues, glucose units. Okay, so that's a glycogen. Here you see glycogen molecule, branching is highly profused here. So and you can see alpha 1,4 linkages as well as 1,6 linkages are present. All these are glucose units. So this is how the weeded structure of glycogen appears. This glycogen, it is again similar to starch, I said. So it is also a rich source of glucose, and that means it is also a rich source of energy to your body. That means when you consume any food item from, particularly when you eat meat and all, you are going to get glycogen and your body generates energy by using that glucose. Not only that, when you eat a lot of food and when they are extra, that means this glucose will be stored in the form of glycogen in your body. Okay, so in our body also glycogen will be stored. Okay, so glucose will be stored in the form of glycogen, and whenever you are hungry, when there are no other sources of glucose, glycogen will be converted into glucose. I think you are familiar about that particular concept. The next type of polysaccharide what we will be discussing is a homopolysaccharide, is a dextran, and these dextrans are actually produced by bacteria and yeast. Okay, and they are formed of alpha 1,6 linkages of glucose. Okay, alpha 1,6 linkages of glucose. Not only that, they'll also have 1,3 branches and sometimes 1,4 branches and 1,2 branches also. So there are different varied number of branching is seen in case of dextran. Okay, in case of glucose and starch, it was alpha 1,4 and alpha 1,6, but here it is 1,6 linkage, 1,3 linkage, 1,2 linkage and 1,4 branches are also found in case of dextran. Okay, and where do you find? Usually we get dental flakes, and these dental flakes are found of these are dextran. Okay, the plaque that is present on your teeth, which is because of certain bacteria, is nothing but the dextran. Not only that, these dextrans are synthetically produced and they are used for different purposes, particularly in these chromatographies, gel exclusion chromatography. In them, okay, so it is used. Cephadex is one of the in what medium. So in that stationary state, so there it is used. Okay. So this is the rough structure of this dextran. You can see alpha 1,6 linkages and alpha 1,3 linkages are present. I think 1,3 linkages are more clear in this particular, in the second diagram. Okay, so let me just show it so that you won't get confused. So this is the alpha 1,6 linkage, and here we have 1,3. See why 1? This is 1 and this is the third carbon. So branching is present here between these two, so it is called as 1,3 linkage. 1,3 linkage. Okay, this one. If the branching is between this and this, then it is 1,3. If it is between this and this, it is 1,2. If it is this and this, then it is 1,4. Okay, so branching, varied branching is seen in case of dextrans. And not only that, I said it is used in gel exclusion chromatography. It can also be, it has medicinal application too. So it is used in antithrombotic, okay, thrombotic, to reduce the blood viscosity, and also expander in case of hypovolemia. Okay, and it is also used in eye drops. Okay, so that's an application, medical application. Now there is one more thing, that's a dextrin. Dextrin is not truly a polysaccharide, it is more a oligosaccharide, and it is formed of only few like monosaccharide units, and it is produced when glycogen is digested, or starch is digested by the help of amylases. We know that alpha amylases cannot break 1,4 linkages, 1,6 linkages. So wherever there is a 1,6 linkage, that will be undigested by the amylases, alpha amylases. So in those areas you find a small chain of carbohydrates. So 1, 2, 3, maybe 4 somewhere here, and then there is a branch like this. So this is what a dextrin is. It might be length of 3 to 10 monosaccharide unit, and it is a byproduct of digestion. So generally called as limited dextrins, and then they will be digested by maltase. Okay, so this is the dextrin structure. They are smaller one and formed of 1,6 bond. What you are seeing here on the picture is actually both dextrin and dextran. Okay, it's a more a dextran diagram rather than a dextrin diagram. So you see 1,3 bond, you see 1,2 bond, you see 1,6 linkages. If you're talking only about this much, then it is a dextrin. Whole of it is a dextran. Okay, yes. So that's all about the homopolysaccharides. Moving on to heteropolysaccharide. So heteropolysaccharides are I think we can continue heteropolysaccharide in another class. So just stop it here with the introduction to heteropolysaccharide. Heteropolysaccharides are they contain 2 or more different kinds of polysaccharide units. Okay, homopolysaccharide contains only one type. Heteropolysaccharide contains more than one type of monosaccharide units. That means on breaking you get more than one type. They can be repetitive disaccharides or repetitive trisaccharides. Okay, and the examples for these heteropolysaccharides are hyaluronic acid, heparin, chondroitin sulfate, and keratin sulfate, and many more anyway. So we will be discussing at least 3 of these in our next class, and that's all for today's class. Thank you.